Good morning, and thank you for standing by, and welcome to the FDA webinar. Your lines have been placed on a listen-only mode until the question and answer segment of today's conference. At that time, you may press star 1 to ask a question. Today's questions will be taken via phone only. Today's call is being recorded. If you have any objections, please disconnect at this time. I would now like to turn the call over to your speaker this morning, Ms. Irene Ihier. Thank you. You may begin.
Thank you. Hello, and welcome to today's FDA webinar. I am Irene Ihier of CDRH's Office of Communication and Education. On October 7, 2016, the U.S. Food and Drug Administration published the final guidance documents, Self-Monitoring Blood Glucose Test Systems for Over-the-Counter Use, and Blood Glucose Monitoring Test Systems for Prescription Point-of-Care Use. The purpose of the final guidance documents is to improve new blood glucose meters by providing FDA's recommendations to industry about the types of information to be included in their pre-market submissions, 510Ks, for these devices. The focus of today's webinar is to share information and share questions and answer questions about the final guidance documents. Today's presenter is Dr. Leslie Landree, Diabetes Team Leader in CDRH's Division of Chemistry and Toxicology Devices in the Office of In Vitro Diagnostics and Radiological Health. Following the presentation, we will open the line for your questions related to the topics in the final guidance documents only. Courtney Elias, Director for the Division of Chemistry and Toxicology Devices, and Ryan Lupert, also from OIR, will join us to assist with the Q&A portion of our webinar. Now, I give you Leslie.
Thank you, Irene, and thank you everybody who's tuned in today to listen to this webinar. The purpose of the webinar is to review the 2 glucose guidance documents that have recently finalized. During the presentation, I will provide an overview of the purpose of the 2 guidance documents, discuss how these documents differ from what we have typically seen in submissions for these types of devices, and highlight the key differences between the final guidances and the draft guidances. Before I get started, I'd just like to review some of the common acronyms that will be used frequently during this presentation. SMBGs is for self-monitoring blood glucose, BGMS, blood glucose monitoring system, OTC, over-the-counter, and POC, point of care. Blood glucose meters are used by very different and unique intended user populations in a variety of settings. They are used by lay users at home. Also, they are used in a variety of healthcare settings, such as hospitals, nursing homes, doctors' offices, among other settings. And as you may know, blood glucose monitoring systems are considered moderate risk and are therefore Class 2 devices that require 510K clearance prior to marketing. During the 510K review, we evaluate the performance of the device for its intended use. However, this is not an independent evaluation of the device. Manufacturers are responsible for the demonstration of the performance. Therefore, the device information and performance data that we evaluate is generated and provided by the manufacturer. For the majority of glucose meters, manufacturers have historically sought over-the-counter clearance, which has typically been done to obtain waiver for these devices so that they can be used in a variety of waived settings. In taking this approach, what has happened is that the majority of meters on the market today, including most of those that are used in professional healthcare settings, including hospitals, were designed and validated only for over-the-counter use. That means that healthcare professional use for these devices was not evaluated. It has become increasingly clear that these different use settings comprise distinct intended use populations with unique characteristics that should be considered during device design and validation. We have heard concern from various stakeholders over the last several years that glucose meters are not adequately evaluated for the way that they are being used, particularly in the hospital setting, and that they have not been evaluated for some of the intended use populations that they are commonly used in. It is our intent in writing these guidance documents that we were to address the issue by creating 2 distinct glucose documents for the different user populations and particular use requirements. These 2 final guidance documents, titled blood glucose monitoring test systems for prescription point-of-care use, and self-monitoring blood glucose test systems for over-the-counter use were finalized and issued last month on October 7th. The final SMBG guidance addresses only those systems that are intended for self-testing by home users. These systems are for single-patient use only. The BGMS guidance addresses those systems for point-of-care use and are intended to be used for assisted testing in professional healthcare settings, such as hospitals, doctors' offices, long-term care facilities. These systems are intended for multiple patient use. The two guidance documents are meant to be a description of FDA's current thinking on the information that manufacturers should submit to FDA for future glucose meter submissions. These guidance documents are not meant to be guidelines or rules for how hospitals, healthcare professionals, or patients should use glucose meters. They are not meant to be rules or guidelines for how laboratories should validate glucose meters. And they are not meant to apply to those meters that are currently on the market, but should be used when making device modifications and preparing future 510K submissions for glucose meters. Many of the aspects of the new guidance documents are similar to what we currently see for these devices, so I won't go into many of these details today. However, there are some areas of the guidances that are new or different from what we typically see for glucose meters, and I've included some of these key differences here in blue. And I'll go into each of these in more detail in the next few slides, but briefly, they include a different accuracy study design and accuracy goals, a modified way of analyzing and reporting interference data, a slight modification to the cleaning and disinfection recommendations that now includes inclusion of the test strip port for cleaning and disinfection robustness, the inclusion of new flex studies, and the inclusion of test strip manufacturing lot release criteria in the pre-market submission. For the most part, the finalized documents did not change much from the draft documents. However, we did make some changes and clarifications based on the comments we received on the draft documents. We received many comments expressing concern and confusion regarding the implications of the guidances on the CLIA waive status of these devices. In response, we added clarifications on the intent of the guidances to the guidances and the implications for CLIA waiver status. Some of the performance goals have also been modified, and the interference testing section was also modified, again, based on the comments we received on the draft documents. What I'd like to do now is to discuss the two documents in a bit more detail, starting with the BGMS guidance for prescription point-of-care use. Then I'll discuss the SMBG guidance in less detail, giving a higher level overview since there's so much overlap between these two documents. The intent of the BGMS guidance is to describe the studies that should be conducted to demonstrate performance for devices that are intended to be used in a diverse professional healthcare setting on subjects in various states of health. Intended use populations for BGMS devices may include patients in all professional healthcare settings, patients in specific healthcare settings such as emergency response vehicles, or patients in long-term care facilities, or patients in a physician's office. This document does not address those systems that are intended for over-the-counter use by lay users at home. These BGMS systems are intended for point-of-care use in specific professional healthcare settings and should be studied in those specific populations and should be labeled appropriately. Performance studies should account for factors such as various disease states, patient conditions, physiological states, and medications that may affect device performance in the intended use population. For those systems intended for use in many or all professional healthcare settings, it may be necessary to identify specific subpopulations in which the BGMS may function differently than in the broader intended use population. These subpopulations and the differences in performance should be identified in the labeling. FDA's clearance of 510K submissions for SMBG devices intended for over-the-counter home use allows for automatic CLIA waive categorization. Now, as discussed in the previous slides, most blood glucose meters on the market today, even those that are used for healthcare professionals, were previously submitted to FDA with claims for over-the-counter use by lay users and were therefore given CLIA waived categorization. Clearance of BGMS systems as prescription use devices intended for point-of-care use in professional healthcare settings as described in this guidance means that they will not be automatically CLIA waived. However, FDA recognizes the importance of having CLIA waived blood glucose meters in point-of-care professional healthcare settings and intends, through the studies described in this guidance, to facilitate CLIA waiver for these devices. The recommendations in the BGMS guidance, particularly the recommendations for the accuracy user studies, are designed to simultaneously support both clearance and CLIA waiver. For instance, the recommended number of samples and the use of the participants in the study that are representative of the intended CLIA waived users will help to support both clearance and waiver. Submitting the information described in the guidance as a dual 510K CLIA waiver submission will help to enable BGMSs to be CLIA waived concurrently with the 510K clearance. So to go into accuracy in more detail, this guidance document describes the accuracy assessment of BGMS systems which will be performed in the hands of the intended users. The study should represent the actual claims made for the device and should include subjects that accurately represent the intended use population. A minimum of 350 patient samples per claim sample type or matrix should be used. Now, this is an increase from what we currently see. But this means that for each claim, such as an arterial, venous, or capillary blood claim, 350 samples for each type should be submitted. So these different sample types can be acquired from the same patient and be used for these different studies. These samples should span the glucose measuring range for each sample type and be compared to a comparator method. If natural samples with concentrations of the extreme ends of the measuring range are not obtained during the study, then additional samples may be contrived by spiking or glycolysis to obtain an additional 50 samples with high glucose concentrations greater than 300 mg per deciliter and 50 samples with low glucose, less than 80 mg per deciliter. Another difference from the studies that we currently see is the recommendation in the guidance that the test strips used in these studies have undergone typical shipping and handling conditions prior to use in the study. If the intended use population of the device includes neonates, then a separate neonatal study should be performed to support this use. Neonatal blood is known to differ from adult blood in that it can often have higher hematocrit levels and lower blood glucose concentrations compared to adult blood. Because these differences may have a direct impact on the performance of the blood glucose monitoring, a neonatal accuracy study should be performed to validate any neonatal claim. Neonatal samples are defined as samples from subjects less than 28 days old. And for this study, the accuracy of 100 to 150 fresh neonatal capillary blood samples should be assessed by comparing meter results to the results from a comparator method. Now, before going into the accuracy goals described in this guidance for the BGMS systems, I'll briefly touch on the 2013 version of ISO 15197. FDA does not recognize this standard, but the glucose guidance documents try to address some of the issues that FDA believes are important for these devices that were not addressed in the standard. By differentiating over-the-counter versus point-of-care healthcare use, these guidance documents address the different user populations for these devices and do not just focus on over-the-counter use. In doing so, the guidances address different use and design requirements important for the different use settings that are not addressed in the standard, as well as assessing the performance in the hands of the different intended users. We have also addressed the need for the improved performance in the hypoglycemic range in the guidance documents. Though not identical to the studies described in the ISO standard, the FDA documents describe studies that are designed to be compatible with ISO, so that studies performed according to the guidance documents will also satisfy ISO. Now, the accuracy goals that are described in the BGMS guidance for point-of-care use have changed slightly since the draft guidance and have been loosened some based on comments we received on the draft. The draft stated that 99% of meter results should be within 10% of the comparator method for glucose concentrations greater than 70 and plus or minus 7 mg per deciliter for glucose concentrations less than 70, and that 100% of values should be within 20%. This final guidance now states that 95% of the meter results should be within 12% for glucose concentrations greater than 75 or within 12 mg per deciliter for concentrations less than 75, and that 98% instead of 100 of the meter results should be within 15% or 15 mg per deciliter of the comparator method. In cases where data points fall outside of these accuracy goals on the previous slide, a clinical justification should be provided in the pre-market submission for each of these data points, along with a description of any other proposed mitigations. And as described in the draft, the study should be accompanied by a description of the study setting, including the size, type, and location of the site should be provided in the pre-market submission, along with a justification for how the study conditions represent the intended use conditions. The patient demographics should be described, including the age, disease states, and any medications that each study subject may be on, along with a description of the shipping and handling conditions of the test strips prior to use in the study. The pre-market submission should also include an evaluation of the effect of potentially interfering endogenous and exogenous substances on the device. This study should include common conditions such as lipemia, varying hematocrit levels, as well as common medications. There are certain substances that we know from experience interfere with current meter technology, such as acetaminophen, uric acid, xylose, etc. However, in addition to testing the common interfering substances, we recommend that manufacturers conduct a risk analysis to assess commonly used drugs in the intended use population. This analysis should take into account device technology, the intended use population, the intended use setting. Any relevant potential interference should be included in the testing. The study design for these studies is not really all that different from what we see now, but there are a few differences. Similar to what we see now, it is recommended that a comparison be made between a test sample that contains the potential interferent measured on the candidate meter and the result of a control sample containing a solvent or vehicle, also measured on the candidate device. The analysis and presentation of the results does differ from how we typically see these studies analyzed. The glucose documents describe presenting the results as relative bias and percent bias with 95% confidence intervals. Now, this is instead of using the more typical non-interfering criteria. Depending on the bias, the interferent, and the concentration of the interferent, language may be added to the labeling to explain the expected device performance in the presence of each interferent. It is well known that hematocrit levels can significantly affect blood glucose meter readings, with a significant positive meter bias at low hematocrit and a significant negative bias at higher hematocrit concentrations. Due to this known interference, a separate hematocrit study should be performed with these devices, as is currently done. The blood samples used should reasonably span the glucose measuring range and the hematocrit range. The meter results should be compared to a comparator method by comparing each meter replicate to the average of the comparator value. The bias for samples with glucose concentrations greater than 75 should be less than 5% on average, with no individual value greater than 10%. For samples with glucose less than 75, the absolute bias should be provided along with 95% confidence intervals and a clinical justification. The guidance states that for point-of-care BGMS systems, a minimum hematocrit range of 10 to 65% should be validated in claims. This is based on the very broad hematocrit range that could realistically be presented in the intended use population for this type of device. If the device is intended to be used in patients with a broad range of blood oxygen levels, then a study should be conducted to demonstrate the range of blood oxygen levels that the device can be used with. The oxygen study may be supplemented with blood oxygen levels of patients used in the method comparison and the user evaluation. If blood oxygen levels do not affect the performance of a particular device, then a comprehensive justification should be provided, supported by analysis of blood oxygen levels on the device performance seen in the method comparison and user evaluation study. As I'm sure most of you listening are aware, that in 2010, FDA modified the way we review all blood glucose monitoring systems to ensure that validated cleaning and disinfection instructions are provided to users. This was in response to concerns that had been raised regarding bloodborne pathogen transmission linked to the shared use of blood glucose meters when appropriate infection control measures had not been followed. This guidance document describes validation studies that are not much different from what manufacturers are currently doing. One difference that I would like to highlight is the expectation that meters be designed such that all external materials can be cleaned and disinfected. This includes the test strip port, other ports, and any seams on the meter, etc. The validation studies in the two guidance documents are almost the same, differing mainly in the number of cleaning and disinfection cycles that represent the amount of cleaning and disinfection that the meter will be exposed to in its use life. Therefore, a meter used in multiple times a day on multiple patients in a professional healthcare setting will need to be robust to many more cleaning disinfection cycles as compared to a single patient over-the-counter meter. The validated cleaning and disinfection instructions should be included in the device labeling. The BGMS guidance also includes a description of flex studies that should be performed, some of which are new from what we can currently see. These studies are designed to demonstrate that the BGMS device design is robust and that all known sources of error are effectively controlled. The test system design should incorporate fail-safe mechanisms whenever it is technically feasible. Examples of fail-safe mechanisms are the use of lockout functions to ensure that the test system provides no result or an error, rather than displaying an incorrect result in cases where, for example, test conditions are inappropriate. Other fail-safe mechanisms are those measures that are put in place to prevent operator error, such as inserting the wrong test strip. The manufacturer should conduct a comprehensive risk analysis that identifies all potential sources of error, including test system failures and operator errors, identifying control measures to reduce the risks of these sources of error, and then, when appropriate, flex studies should be performed to validate the effectiveness of the control measures. This guidance document includes several examples of such flex studies, and some of them are listed here. So I'll highlight just a couple. Test strip stability should be established by assessing test strip performance throughout the claimed shelf life and conditions. The effects of temperature and humidity during device use should also be assessed. The use of used test strips or insufficient blood sample volume should also be assessed. Included in the guidance that is new from what we currently see is a request that the description of the test strip manufacturing lot release criteria be provided in the pre-market submission. Lot release criteria is criteria used by the manufacturer to assess and release into market manufactured lots or batches of test strips. This has been included in the document to try to help prevent less accurate test strips from being released onto the market. Test strip lot release criteria should be sufficient to ensure consistent quality of test strips. They should be designed to ensure that release lots conform to the labeled device performance in the hands of the intended users. These criteria should be more stringent than the criteria used to evaluate total error in the performance studies. And these criteria should assure statistical confidence for a full lot. Manufacturers should provide a description of the lot release criteria and a summary of the sampling scheme and acceptance criteria in the pre-market submission. Now, I'll discuss the SMBG guidance briefly to highlight some of the key differences. This guidance is meant to address only those blood glucose monitoring systems intended for lay users at home. This encompasses individuals with wide ranges of age, dexterity, vision, and training received on performing the testing, and other factors that can be critical in the patient's ability to accurately use the device and interpret test results. SMBG systems should be designed to be robust and reliable to accommodate actual use by people affected by diabetes, such as more varied storage and handling conditions as compared to devices used in professional settings. This document is not meant to address those systems that are used for point-of-care use. As in the BGMS guidance, this document recommends that accuracy of the system be demonstrated in the hands of the intended user, which in this case is lay users. And this should be done under conditions reflective of expected use. The recommendation is to use a minimum of 350 patient samples obtained and tested by the lay users when they are given only the device and the device labeling with no training, coaching, or prompting. 350 samples should be obtained per claim sample type, such as for fingerstick, palm, or thigh. These samples should span the glucose measuring range for each claim sample type, and the meter results should be compared to the comparator method. At least 10% of the participants in this study should have no experience with SMBGs. Now, these subjects can be non-diabetics. Test strips used in the study should have undergone typical shipping and handling conditions prior to the study. We recognize that the user study may not provide a sufficient evaluation of the device performance of the extreme upper and lower ends of the measuring range. Therefore, if natural patient samples are not obtained at the extreme ends of the measuring range, then additional samples may be contrived by spiking or glycolysis to obtain an additional 50 samples with high glucose concentrations greater than 250 milligrams per deciliter and 50 samples with low glucose concentrations less than 80 milligrams per deciliter. The accuracy goals outlined in the SMBG guidance have not changed from the draft. And they stated that 95% of SMBG results in the accuracy study should be within 15% of the comparator method across the entire claimed measuring range of the device, and that 99% of the SMBG results should be within 20% of the comparator method. If there are outliers that do not meet the accuracy goal on the previous slide, they should be specifically addressed by manufacturers in the pre-market submission with a justification for why the errors occurred and a description of the potential for that error to affect user safety when extrapolated to the intended use setting, that is, when millions of test strips are used. We understand that some SMBG devices may not be able to be to reliably measure glucose at very low concentrations and meet this accuracy goal. However, we've heard from the clinical community that it could be acceptable for lay use to narrow the claimed measuring range based on the accuracy results so that readings as low as 20 milligrams per deciliter are not reported for a particular device. However, the measuring range for this intended use population should not be any more narrow than 50 to 400 milligrams per deciliter. The interference testing described in this document is the same study design as outlined in the BGMS guidance, with the testing of common endogenous and exogenous substances that may interfere with the technology and that would likely be present in the user population. As with the BGMS document, the meter results from the test sample are compared to those of a control sample. And the relative bias and percent bias with 95% confidence intervals are provided instead of the non-interference criteria. As with the BGMS document, the effect of hematocrit on device performance should also be evaluated. However, here a claimed hematocrit range of 20 to 60 is considered ideal, since the intended user population may reasonably be expected to have hematocrit values between 20 and 60%. Now this document also lists a minimum claimed range that should be no narrower than 30 to 55% hematocrit. And in this SMBG document, the goal is to have a bias of less than 8% on average for the study for glucose concentrations greater than 75, with no individual value having greater than 15% relative to the comparator. Similar, the infection control studies are very similar in this document as described in the BGMS document. The validation studies are based on expected use and the use life of the device. Therefore, the single patient use devices will typically have fewer validation cycles for the robustness study to support the device use life. But as with the BGMS guidance, the over-the-counter meters should be designed so that all external materials can be cleaned and disinfected, and this includes the test report. The validated cleaning and disinfection instructions should be included in the labeling in language that is easily understood by home users. The SMBG guidance recommends similar flex studies that are described in the BGM guidance, and also recommends that the test strip manufacturing lot release criteria be provided for the over-the-counter systems. For SMBG devices, the following warning should be included prominently on the outer box label and package insert for SMBG systems. This warning emphasizes that the system is not intended for use in healthcare or assisted use settings such as hospitals, physician offices, or long-term care facilities, since it has not been cleared for these uses, and the use on multiple patients could lead to the transmission of bloodborne pathogens such as hepatitis B. Currently, consumers have no way to distinguish meters based on performance before they purchase them, so that lay users, along with their healthcare professionals, have the ability to choose the SMBG that is right for them. It is important to clearly describe the accuracy of the device in a way that is easy for them to understand. It is also important for this information to be located in a prominent place in the product labeling so that lay users can understand the performance prior to purchase and also when they are learning to use the device. Therefore, the SMBG guidance recommends that accuracy information be placed prominently on the outer box label and in the test strip package insert. The guidance recommends that the data be presented in easily understandable tabular as well as graphical formats. The tabular example that is provided in the guidance is similar to the accuracy tables currently used for these devices, with the percentage of meter results displayed within different accuracy bins, such as 5, 10, 15% of the comparator method. This is the example of the tabular format that's included in the guidance. Here the numbers represent the number of meter results that were within the level of accuracy shown relative to a laboratory comparator method. Now these are only examples, we continue to encourage manufacturers to discuss alternate ideas with us. Now that the guidances have finalized, we're working with manufacturers on phasing in the recommendations with the goal of phasing them in over the next several months. Now, we recognize that the development of the current devices were performed prior to the release of the guidances, but we encourage manufacturers to contact us with questions and concerns with implementation. We have been contacted by manufacturers that are in situations where they have completed their studies and are prepared to submit their submissions when the guidance was published. If you are in a similar situation or have other questions or concerns, we encourage you to contact us. We also recognize that certain recommendations may be easier to implement, such as the labeling recommendations, as opposed to other aspects of the documents. And that concludes my slides, and we'll be taking any questions you have now.
Thank you. At this time, if you do have any questions or comments, you may press star 1. Please unmute your phones and state your first and last name when prompted. Again, that is star 1 if you do have any questions. One moment, please. David Phipps, you may go ahead.
Good morning. I just have a question. I may be a little bit off subject. LifeScan, who, as you know, makes one-touch meters, is telling people from their customer service end that FDA allows a 30% difference between differing brands of monitors and a 20% difference between your actual blood work, what it shows. I have a patient who recently, they had a Walgreens TrueTouch meter that was made by Nipro, and those were discontinued. He had some strips left. His insurance company recommended, I guess the preferred was a one-touch mini. So he got that and started noticing that, you know, he's testing 100 on the TrueTouch and he's testing 100 to 30 and 140 on the Ultra Mini. So when he called him and asked him about it, he was told this, and then he kind of, you know, he argued with him about it. So well, he never heard that, and his doctor never heard of that. He provided them with the last 2 weeks of values on both of the meters, and they agreed that he was running above 30%. I think he was running around 33, 34%. So they sent him a new meter, which basically has the same results. So when he went to talk to his doctor, I mean, you know, if he got a 90 on his new meter, he's actually testing around 65, 68 on his TrueTouch and wondering why he was feeling dizzy. Question I have is if that's actually a standard, and we use these meters in the hospital, you know, it makes it kind of unusable for a sliding scale. You know, if you've got one value versus another. And actually, this patient had come back and told me that his values were actually closer with the TrueTouch to his lab work that was drawn in the doctor's office than the One Touch was. But of course, One Touch, like all the rest of them, claims that theirs is the most accurate on the market. Is this going to be, number one, are those values true as far as how much the FDA allows currently, and is this something that's going to be corrected by the 95% that should be within 12%?
Hi, yes, this is Leslie. Thank you very much for that question. We understand exactly what you're saying, but the criteria that we have, we don't compare meter to meter. We don't have any sort of acceptable difference for that.
Okay.
You're correct. The goals of these guidances really are to improve the consistency and quality for glucose meters in the different intended use populations.
Sure. The other concern I have is that he's not on insulin, but if were he on insulin, he'd be giving himself more insulin, you know, based on these higher values. And I think the public at large has been, certainly as a healthcare professional, I've always been given to believe that as long as you're using their strips and the proper technique, that your accuracy should be basically the same from one meter to another. I had no idea that there was that much of swing that was allowed. Is that true, though? Is that 30% difference that they say is allowed by the FDA currently? Is that accurate?
No, we don't have any such 30% criteria. And I hear some concerns from you based on the results that you've seen. And as always, if you have any concerns, we always encourage you to submit an end-watch report if you have any problems with any devices. Okay.
That was my next question, was who goes back and talks to them for making these kinds of claims? You know, I mean, they're dragging you into this and basically saying the FDA says we're allowed to be this far, or we're allowed to have this much of a difference between two different brands of meters.
Sure. We review all those reports and try to address them appropriately.
Okay, great. Thank you for the information.
Thank you. Our next question comes from Nicole Crumpt from Lab Services. You may go ahead.
For the infection control, the cleaning and disinfection, should the entire device be used for the cleaning and disinfection testing, or can the individual materials from the device be tested?
Right. Hi, this is Leslie again. There are two aspects to that testing. For the disinfection efficacy, where you're demonstrating that the disinfectant is effective against hepatitis B, you can use parts of the meter. So it's called coupons. So every external part, like the buttons, the display, the case, can be used. But you don't have to use the complete full device. But for the robustness testing, which is the second component to the testing, the entire complete device should be used.
Okay, thank you. I have one follow-up question then. So the pieces of the device and the materials, should that be, can they be new materials, or should they be the materials that were used in the robustness testing?
Oh, no. Typically, they're new materials.
All right, thank you.
Thank you. Once again, if you do have any questions or comments, please press *1. Please unmute your phones and state your first and last name when prompted. Michael Chabero, you may ask your question from Limco Health.
Hi there. So one of the questions we have is, how does the guidance document play into a special 510K for a currently cleared device? If we're submitting a special 510K, do we have to comply with the new guidance, or will it be assessed against the guidance and standards in place when it was cleared?
Hi, this is Courtney Elias. So guidance documents really are meant to outline the types of studies that we think that manufacturers should do to address certain risks or performance evaluations for that particular type of device. And the type of submission doesn't actually matter with respect to a particular study. So if for the change you're making, one of your verification and validation activities requires some sort of study that you do to address the risk you've identified, we would recommend you follow the guidance document in performing that study. But, as you know, for a special 510K, if you're modifying a cleared product, you would do a risk analysis to identify the types of verification and validation activities that are necessary for that change. So, like I said, when you're doing a study that's addressed by the guidance as part of your V&V activities, we do recommend you follow the guidance. Certainly there are other activities that are V&V activities that may not be addressed by the guidance also. And we're also happy to help with any questions on particular situations that you have that might be unusual as well.
Okay, great. I have one other question as well, if that's okay. Different topic. So when we are doing the accuracy studies, the guidance document just says that all data must meet those 95% and 99% requirements. So, but you're using a comparator. Is it okay to essentially lump all the data together for that assessment, or should it be assessed at each comparator value?
I'm not sure I understand your question. Are you saying, what are you saying you'd lump together?
Well, say that I test against a comparator at 30, 50, 75, 100, on milligrams per deciliter. Should the comparison be made at each one of those comparator values, or can I put all that data that I collected together and say, well, I meet it 95% and 99% of the time?
When you're doing the analysis each patient result should be compared to the respective comparator result. When you go to analyze it and present it, the tables are combined for the different glucose ranges, but each individual result should be compared to a comparator method result. Does that answer your question?
Not really. Maybe this is something that I need to take up on an individual basis or with you directly.
I mean, the method you would use to do this is not different than the method that you use now to do it. So for each person that's in your study, you take a meter measurement and a comparator measurement. You assess the difference between those values, and then that difference is what, you look at to see, how many of those subjects have a difference greater than or less than, the goal.
So I guess, say I tested 1,000 subjects, just to make it easy. Then I could just say for all those 1,000 subjects, we met the 95% and 99% requirements.
Yes, if I'm understanding your question correctly, but I'm not sure that I am. So maybe it would be good for you to follow us up on that. But I do want to emphasize this is not different than the way that people typically analyze the data for their current submissions.
Okay, great. Thank you.
Once again, if you do have any questions or comments, please press star 1. At this time, I am showing no further questions. Again, that is star 1 if you do have any questions.
Operator, are there any more questions?
At this time, I'm showing no further questions.
Okay, thank you. Thank you. This is Irene here, and we do appreciate your participation and thoughtful questions. Today's presentation and transcript will be made available on the CDRH Learn webpage at www. Oh, seems like we have a few questions.
We do. One moment, please.
Okay.
Jenny Kim, you may go ahead. I'm not sure what the company was.
Yep, we're from Arcray. And we're calling because we have a question regarding the control solution with every strip. Could you elaborate more on that requirement in the guidance?
Yes, in the public meeting, we received feedback that it would be helpful if when users purchased their meters and test strips, if they had small amounts of control solutions to use for their testing. So the guidance is intended to address that by suggesting that glucose control solutions be included in each test strip vial package.
Would you like to go to the next question?
Yes, please.
Susan Imboden from Alkalite, you may go ahead.
Hi, yes, a follow-up to what was just asked. When the control solution is in each vial package with the test strips, will they test one strip from each of the vials or each of the bottles?
This is Courtney Elias. I think probably we believe that most manufacturers will choose to put a small, sort of smaller than the typical current vials amount in the vial with the test strips. And manufacturers don't typically now tell users how many control solutions to do, but they do make recommendations in the labeling about controlled testing. So manufacturers should assess what type of instructions they should provide to users, and we do recognize that control materials are also available for users who may want to test more often.
Okay, I just wasn't sure what to tell the clients when they ask how often they should test with their control solution.
Right. The guidance document doesn't recommend any changes to the instructions. The concern, I believe, was with the availability of control material for users to purchase new test strips. Right.
Okay, and then my second question is, this is going to be for name brand meters as well as drugstore meters, generic meters?
Yes, all.
Okay, so all of them will have the accuracy requirement on their boxes?
Moving forward.
Yes. Okay, thank you.
Thank you. Dennis Gunault from EPAC, you may go ahead. Sir, your line is open. Dennis Gunault. Once again, if you do have any questions, please press *1.
We'll give it a few moments to have people log in if there are any questions.
One moment. Charles Ryan from Essentia, you may go ahead, sir.
Yes, hi. This is a follow-up to the control solution discussion. I was just wondering if FDA would allow entertaining an alternative, such as providing like a coupon in with product rather than the physical control solution.
This is Courtney Elias. The concern that we heard at the public meeting was related to individuals who wanted to do control testing and also who may be unaware of necessity for control testing but did not have within expiry control materials available to them. So, you know, we're definitely happy to address or to discuss things that may be alternatives to address the issues that we discussed in the guidance document. So we're happy to discuss that with you. We would want to understand, you know, how the proposal that you have, like a coupon, would address that type of concern. So, you know, we're happy to address that. I think that, you know, they would still not have the control material there to test, you know, with a coupon. They'd still have to take an action to order it. But I just wanted to clarify the concern that we were trying to address there. Does that help?
Yeah, I think there's ways that we could still arrange that, but okay, thank you.
Sure, we're happy to discuss them.
Thank you. Dennis Donald, you may ask your question. Please state your company name.
Hi, I think it's Bennett Dunlap, Diabetes Patient Advocacy Coalition.
Caller, are you there?
Hello?
Hi, caller. We lost you for a second. Can you repeat your question?
Hi, Bennett Dunlap. Can you hear me now?
Yes, we can hear you.
I feel like I'm on a cellular commercial. Thank you. It's Bennett Dunlap. I'm with the Diabetes Patient Advocacy Coalition. In the guidance process, patients had expressed concerns both for the over-the-counter and the point-of-care use devices. I think it's interesting that the first question we had today was apparently a healthcare professional that was concerned about over-the-counter devices being used in clinical settings, and I'm curious as to what processes there will be to enforce that the correct device is used in the correct setting, and in the concern over transmission of infection and cleaning, if there will be consideration of vectors other than the actual device, such as the supplies, because there's evidence that suggests that supplies can be a vector for transmission of disease. In the over-the-counter area, labeling, I think, is something that patients were very concerned about in the guidance. And we noted a few years ago that FDA used guidance to say that a device was not approved for care adjustments and not approved to do calibration of the CGM. And I'm curious if FDA is open to definitive labeling claims by manufacturers that a device is approved to make therapy adjustments and is approved to calibrate a CGM, so that we can clearly differentiate devices that are approved under this new guidance from devices that are grandfathered from previous generations of guidance. Thank you.
Hi, Bennett. This is Courtney. I think there's a few questions in there. Let me try to get some of them, and then you can let me know if I missed any. First, you expressed some concern about the other products that people use to test and whether or not they may be vectors for infection. And of course, other products, other types of supplies that people with diabetes use have some of the same issues, but cleaning, disinfection is necessary for all types of medical devices that are blood contacting. So though these guidance documents address blood glucose meters and those products, there are other parts of the FDA that address cleaning and disinfection requirements for other types of products. So it's definitely a concern, and we hope that the efforts that are underway to address those for all products are effective.
Yeah, I guess, Courtney, what I really meant was the supplies that travel with the glucose meter. So if a can of strips is moving from hospital room to hospital room to hospital room and a gloved hand is going into that can of strips or onto that container of strips, that container of strips then can become the vector.
Yes, certainly. That is a question we do ask when we look at meters designed for this type of use. Some manufacturers have chosen individually packaged strips and things like that, which do help.
Yeah, I guess it's the transmission of of supplies from room to room to room. It doesn't matter if they're in a can or individually stripped or whatever. If it's not disinfected, it's just like the device itself.
Right. I also have heard of some healthcare facilities that don't transfer strip files across rooms in an effort to control some of this as well, which is probably good practice. One of the other questions that I think I heard in there, and I may have forgotten a few now, but one of the other ones is that you were wondering about the separation between over-the-counter products and products intended for use in healthcare facilities, more in prescription use settings. This guidance specifies that, if you have a product that's designed for use in a prescription use setting, that it should be labeled appropriately for that use. The labeling will be specific about what the device has been cleared for, what types of assessments have done, and what the limitations of the device are. And, the control that's in place there, I believe, is largely going to be CLIA, and in that the labeling statements that are made are taken quite seriously by CLIA investigators or their the other bodies like CAP, Joint Commission, et cetera, that look at these products in the healthcare setting. For example, if there's a recommendation in the labeling, they really take that as sort of a requirement. So if it's recommended that products are handled in a certain way and a facility is not doing that, they can be cited under CLIA. If there is an intended use population that's not cleared and that's being used in that intended use setting under CLIA, they will have to take appropriate steps to control the risks or do their own validations and take their own responsibilities for that. So with the prescription use settings, there are definitely some controls in place that are intended to help manage that. Not sure I got all your questions. Can you repeat them?
Sure. So that was really great on the OTC side. I guess the question then becomes on the over-the-counter sign, is there a similar process to, not to draw too tight a parallel, but to CLIA, where the recommendations for labeling become something that would be followed by third-party payers? And my concern is that there's a definitive difference between new, more accurate devices, using those to dose insulin, make therapy adjustments, versus devices that, as you said in the past, if once it's approved, it's approved. So we could have technology from the 80s or earlier that's still approved. Well, how do we differentiate with a label that these new devices are approved to make therapy adjustments and they are approved to properly manage diabetes with dangerous drugs like insulin and are approved to calibrate a CGM?
Sure. You're raising important questions that we are definitely discussing with patient community and healthcare provider community that can't be addressed by guidance documents, unfortunately. They probably would require a larger change in order to make other types of requirements apparent for these, but we are looking at other areas. These guidance documents are not the only effort that FDA has in order to try and increase transparency and consistency with glucose meters in a way that benefits all stakeholders. We
Can the
hear about a
label
lot of
be
the
used to do
concerns.
that?
Excuse me?
Can the label be used to do that?
So
sort of the positive version of the negative 2013 label? Right.
So the label, so for example, in the over-the-counter guidance, there are labeling statements that we're asking people put in that will help. So the warnings about use on single patient use only, if you're in a healthcare facility and, for example, the state health inspectors find that you're using them on multiple patients, I think that there would be some liability on that facility for doing that without proper controls themselves. So I personally believe that even those warnings will help some of those situations. With respect to designating certain meters for treatment decisions and other meters not for treatment decisions, we don't have a plan to go in that direction because of the potential, one, we only have one category of glucose meters at the moment, so we would need a regulatory change to create different categories of meters. But we are discussing, different ways of moving forward, and we're happy to hear feedback about what types of needs are still there that will need to be addressed in the future.
Thanks.
Welcome.
Thank you. Once again, if you do have any questions or comments, please press star 1. Again, that is star 1 if you do have any questions or comments. Charles Ryan, you may ask your question. Please state your company name.
Sensia.
Oh, hi.
I'm sorry. Just with respect to the OTC guidance and the accuracy criteria there, could you just confirm that the intent is that the criterion of the 95% and 99% ranges would apply not just to the fingertip results, but also to alternative site testing?
Yes, that's the intent.
Okay, thank you.
And at this time, I am showing no further questions. I'll turn the call back over to Ms. Irene Ihier.
Thank you. This is Irene Ihier, and we appreciate your participation and thoughtful questions. Today's presentation and transcript will be made available on the CDRH Learn webpage at www.fda.gov/training/CDRH Learn by Wednesday, November 30th. If you have additional questions about the final guidance documents, please use the contact information provided at the end of this slide presentation. As always, we appreciate your feedback. Again, thank you for participating, and this concludes today's webinar.
And thank you. You may go ahead and disconnect at this time. This does conclude today's conference call.